Hello, everyone, and welcome to the FOXG1 Research Foundation Science Q&A. As many parents received an email and social media posts saying, we know that you have many questions about the research underway for FOXG1 syndrome, and we want to answer them. So many parents have emailed us their questions, and we are here today to get through as many as possible. And we are joined today by our Chief Drug Development Officer, Dr. Gai Ayalon, and our CEO and co-founder, Nasha Fitter, and our Clinical Research Director, Elli Brimble. So we are going to get started because there are a lot of questions to get through and do our best to answer your questions. And of course, always reach out to us anytime, email us if you have follow-up questions. So let's start. So what I will say is that there were many similar questions about our gene therapy program, about clinical trials, about clinical trial design. So we will put them together. So do note if you don't see your exact question answered, it's because it was already answered from the question being asked by many parents. And the one other thing before we get started is that in November, we are having our FOXG1 Parents Conference, and we will really dig into our state of the union of our research at that time. That's also months away from now, so we will have more data and we will have more to talk about. So we hope that you'll join us in Florida and be together to really talk about everything going on for FOXG1 syndrome. Okay, so let's get started. The first question, and this is a great to open with, and this was from a FOXG1 grandmother who talked about in her recent Race for a Cure fundraiser, so many of your family and friends jumped at the chance to donate in honor of your Fox. And you want to provide them with an update, which I think is fantastic. So why don't we have Guy, actually, I'm sorry, let's Elli put together some slides to give a more common language update on the language that you read on our website. So do you want to pull that up, Elli?
Of course. This was such a fantastic idea. Again, thank you for thinking of it. We will happily make this slide available to everyone so that you can communicate the great work that our community is doing to advance therapeutic options for FOXG1. So I wanted to divide it into 4 sections that I think do a good job at encompassing all of the work that we have going on, which is a lot. And so starting in the first column, here is a summary of the work that Dr. Gai has been leading in our gene therapy development. And we're at a really exciting moment when we're transitioning away from some of the basic science work, moving into our preclinical studies, meaning that we're starting to pull together the critical research needed to begin our interactions with the FDA and other regulatory agencies. So some of the work that has been done so far is looking in animal models and other cell lines to support these upcoming interactions with the FDA and to make sure that the data we provide to them is very solid to demonstrate that we are confident as we can be at this time in the safety and efficacy of drugs before we move into clinical trials. We're also doing some work to investigate other therapeutic strategies beyond gene therapy. And so these have been ongoing to explore these treatment options, looking at programs like antisense oligonucleotides and also drug repurposing screens as well. We have had a very busy past few months in the clinical research space. And by clinical research, I mean focusing more on studies that involve humans and their data. So importantly, we launched a biobank with Coriell, which will allow us to collect samples from individuals and their family members. We also joined and completed enrollment for a study with Combined Brain that is focused on biomarker detection, which can be very helpful in clinical trials as well. As some of you may know and have participated in, we've had a number of interviews conducted to help support our disease concepts model for FOXG1 syndrome, which will help us explain to agencies and others in our community what it means to live with FOXG1 syndrome. Those interviews have been completed, and we look forward to sharing the results with you as soon as we're able to. And then very excitingly, we have also just launched a research study, which we will be sharing more about soon, looking to better understand the frequency of mosaicism in FOXG1 syndrome and the impact that that has on clinical features. And then lastly, we continue to work towards sharing the information that we are developing as part of our research strategy with the broader community. We have 3 full-length research papers that are currently in development and are close to being reviewed or accepted for publication. We also recently presented 3 posters at a gene therapy conference in May, all of which had the great fortune of attending and seeing live, which was fantastic. And then lastly, as Nicole mentioned, in November, we will have our FOXG1 Research Conference. And so many folks are currently busy planning for that. So I hope this gives a good overview that can help you communicate with your families and support systems in the community. And this will be available as a resource for you moving forward.
Yeah, and I'll just add that, getting to a therapy is much more than just seeing something in a mouse model that works. And that, that's how we start. But as Elli has gone through, there's so much other work that we're doing on the clinical side to really partner with the science that we have. And that is necessary for the FDA or any other agency to approve a clinical trial to start. And so that's all the work that is undergoing. It's a lot of work. It's very intricate. And so people that have donated should feel really good that we are doing this work. We know how to do it. We have the right team guiding us that has done work like this for biotech companies and pharma companies and taken drugs to market. And that's the stage that we're in.
Great. Thank you so much for that. That was a great slide and great overview, Elli. So a question from the same grandma. I, like most with loved ones diagnosed with FOXG1 syndrome, have high hopes that this gene therapy that we're speaking about will be ready and a match for our Fox. So to help with anticipation, what are you seeing as best fit potential candidates, age, severity, ethnicity, location? So basically this question is about criteria and clinical trial design, which many parents asked about. So Guy, why don't you speak to that?
Sure, thanks, Nicole. So as you mentioned, this question understandably comes from multiple parents and is clearly front and center. And I think I'll start by saying that developing the clinical strategy as a process, we're now in the exciting time of transitioning into it. There's still a lot of work ahead of us. So we will have some answers. We will not have all the answers yet because some of the answers evolve with time as we have conversations, both with clinicians as well as with the regulatory agencies. All these help shape the ultimate design of a future clinical trial. And this is sort of that phase that we're entering at this point in time. Our gene therapy program is designed to have the potential to treat the broad range of types of FOXG1 mutations. So, and that would include truncations, missense, nonsense, frameshift variants. And in animal models, we've seen efficacy in different types of models of different types of mutations. And all this is through really incredible and admirable work that has been and is being done at the Lee Lab under the helm of Sue and Jay Lee in their group at the University of Buffalo. And I'd say that all this work has really brought us to the point where we are confident in the readiness of the gene therapy program to enter the next phase of toxicology studies. And those are, of course, the gateway to get to clinical trials. At this point in time, it is too early to define what we call the inclusion criteria in terms of what exactly are the characteristics or of the patients that can be enrolled into the study. Those are going to be incredibly important topics that will be discussed with clinicians as well as the FDA and other regulatory agencies outside of the US, starting in the coming weeks, actually, and carrying into 2025. And will ultimately culminate as we get closer to filing an IND, where the entire scope of the clinical trial design will be delineated, and this will need to be vetted by the regulatory agencies. So there's a combination, there's a process here of dialogue with clinicians, scientists, and regulators to ultimately land on the study design, which will include inclusion criteria. So we don't have these details yet.
Thanks, Guy. Yeah, that's a big question. You know, we're working on this gene therapy, and parents want to know, you know, the things that we just don't know yet, but we will know, and that's all part of this very big process. Okay, the next question is from a mom in Sweden who, reading our Q2 research update, she was excited to learn about our gene therapy program, and saw something we had in there that FOXG1 Research Foundation Australia is preparing for clinical trial readiness with the Sydney Children's Hospital. And her question was, is this something she should try to start up in Sweden? And so we can definitely speak to that as well. Guy, did you want to? I know we had talked about that briefly. So why don't you share your thoughts?
Right. So ultimately, our goal is to have an approved treatment for FOXG1 syndrome that would be accessible and available to all patients, and try to do this as early as possible. Now, clinical trials are an important step on the way to an approved drug if the drug is successful in clinical trials. So I think that this is important because we will not be able to run trials everywhere, right? So the idea is to run trials at the most efficient way, such that we can reach a conclusion as to whether the drug is working, and if it is working, to get it approved such that it will have broad distribution to all patients. How we decide about the locations of clinical trials is part of the process that we will be embarking toward the end of this year and into 2025. And it is a complex decision that has to do with logistical and regulatory and budgetary considerations. The FOXG1 Research Foundation will be conducting an in-depth sort of feasibility assessment next year to chart the optimal number of sites and their locations for clinical trials. Our Australian chapter and its CEO, Steve Hill, is part of an initiative in Australia that is looking to set up a whole infrastructure to run clinical trials for rare diseases. This is sort of beyond even the scope of FOXG1. It's a bigger effort. And of course, that gives us an opportunity to also conduct direct feasibility for FOXG1 trials in Australia. For the EU, we will definitely be exploring clinical trial sites in Europe. The regulatory path in Europe is different and therefore requires a slightly different approach. And we will be working with clinical operations and regulatory consultants in order to identify the countries and sites within Europe that would help us streamline the clinical development strategy as quickly as possible. So I would say that specifically to the question of setting up activities in Sweden, I would say it's premature at this time. We first need to do the feasibility in terms of finding the best countries to conduct trials at least the first pass of trials. And then once we have those countries identified, then we can trigger specific activities for these countries.
Great. And then this is a very common question is, will the gene therapy be a one-time injection? Like, how does a gene therapy actually work?
Yeah. So this is a great question. And at this point in time, the community, the scientific, the medical community is seeing AV9 gene therapies as what is called one and done. This is certainly the hope. What is important to recognize is that gene therapies as therapeutic approaches are relatively new, which means that even the ones that are ahead of us have not been in the clinic for a very long period of time. So the understanding of how these therapies last from a treatment duration perspective over years and decades, this information is not yet available. The hypothesis is that it will be long lasting and maybe one and done. And with time, we will learn as a community, as a clinical and scientific community, whether this is indeed the case. So for now, we are operating under the assumption that it is one and done. That being said, we will learn, of course, from programs for other conditions that are ahead of us, as well as learn from our own programs. And importantly, as a foundation, we're continuing to pursue the new technologies and new approaches all the time, such that if and when there is a need for something else or something better or an enhancement, that we are on the cutting edge of the therapeutic research and efforts to enable that.
Yeah, that's a great point. We can't emphasize enough that, as you know, the FOXG1 Research Foundation is this global organization, and we are at the beginning of this genetic science revolution, right? I mean, we're seeing a lot of it every year. It's just exploding, but it's still at the beginning of it. So, we're going to continue. This is going to go beyond us. We're really building the foundation of the foundation. So, this is something, well, actually, that actually brings me to another question, and I'm going to jump ahead in our questions. Are all efforts currently focused on gene therapy? And Elli spoke to that a little bit, but we should speak a little bit more to that.
Right. So the answer is that no, not all efforts are funneled only to the gene therapy. We believe that our gene therapy approach is, at this point in time, with the available sort of most advanced technology out there, is the approach that has the highest probability of treating as many different FOXG1 patients or the broader patient population. That being said, we are continuing to pursue other approaches, whether those are antisense oligonucleotides, gene editing, tRNA, small molecule repurposing, and all these are continuing and will continue, and we will continue to add to them as we learn and have the opportunities to explore additional new technologies that are emerging. We've seen exciting things coming up at ASGCT in Baltimore last month, as Elli mentioned. And we will continue to assess the readiness of these technologies to be sort of imported, if you will, into us through collaborations with other labs and other companies. It is, however, also important to mention that all these require resources, and ultimately our ability to move fast with additional programs and technologies really depends on the resources that are available to us. Naturally, we want to get the gene therapy to the clinic as quickly as possible. This is our main effort at this point in time. And then the more we can do with additional resources, we will.
Great. Okay, so actually another question from the same mom of a 3-year-old boy who asked about all our efforts currently focused on gene therapy. She also asked, when do you expect clinical trials to begin for our gene therapy program?
Right. So this is what we can say at this point in time is that we can provide an estimate, which I will. It is, however, important to put the caveats around the estimate because the readiness for clinical trial requires alignment, of course, with regulatory agencies and is dependent on the results of the toxicology studies. So what I would say is that if things go well, I foresee us being able to initiate clinical trials as early 2026. We are embarking on our toxicology studies later this year, 2024, which will carry into deep into 2025. And then based on the results of these studies, we will have our regulatory interactions with the various agencies, and assuming all goes well, early 2026, we hope to initiate clinical trials in human patients.
Nasha, any thoughts on?
I mean, we've been waiting a long time for that, and it gives me goosebumps to think that it is so close, even though as parents, of course, every day feels really far, but we are talking less than 2 years. And so much of what Guy said, it is so critical. There's so much worry about AAV9 that it is a one-time treatment, which is great in many ways, right? You're one and done. The other aspect is, all right, I've got this one-time treatment, and if it doesn't work perfectly, can I do anything else? And yes, you can't get another AAV9 treatment, but you can get other treatments. And there are more and more interesting treatments coming down the pipeline every single year. So I think that fear is, it makes sense, but there's a lot of hope that even if this treatment doesn't work perfectly, there's a lot more things that you can do. And also with clinical trials, we're so hung up on clinical trials, we just want them to start. But if your child does not get into the clinical trial, it's okay, because our goal as a foundation is not just to help the kids, the 10 or 15 kids in the trial, it's to really get the drug approved for every child with FOXG1 syndrome. And we're going to be racing to do that as fast as possible.
It's a really key point, because it is so much of the work is about, getting access to all the children. Guy and I had a call earlier where we're talking to a parent about the mission of the FOXG1 Research Foundation since we started is to be here for all children, all mutation types, anywhere in the world. That is our mission. And that's really important because there are different mutation types, and one therapeutic might not work for another. So as an organization, we are, it is our onus to make sure that we are exploring all avenues and understanding deeply all the different mutation categories of FOXG1. Yeah. Which Elli very much understands the different mutation categories of FOXG1. Thank you for all you do. There was a question here from the same mom who what she's asked is, I'd like to know which cell lines you included in our gene therapy. So I think maybe Guy, a little explanation has to go into how gene therapy works a little bit more to answer which cell lines you guys included in the gene therapy.
Right. So I think that cell lines aren't necessarily included in a gene therapy. So maybe here a small explanation will help.
I think if I can read between the lines,
Please.
this parent question might be more about, is the gene therapy only going to work on certain mutations? And those are the cell lines that we experimented on.
Right. So I would say a few things. So since FOXG1 syndrome is a haploinsufficiency, which means that the level of expression of FOXG1 protein is lower than normal or neurotypical, then the goal is to increase the expression of FOXG1 protein in the brain. This is ultimately what the gene therapy is designed to do. That's the approach that we're taking, as well as I'm sure other similar efforts for gene therapy for FOXG1. And there are different experimental tools to assess whether your candidate therapeutic is increasing expression levels of FOXG1, and one of them is to use cell lines, and the other is to use mouse models, just as an example. So for our gene therapy candidate, this gene therapy candidate has been tested in multiple lines and in multiple mouse models, in a mouse model of truncation, of missense, and nonsense mutations. And so we have our approach is to design a therapy that would apply to the broader patient population as much as possible. And our preclinical testing has indeed demonstrated that it is applicable across all these various mutation types. Now, you can do this in cells, you can do it in animals, but to your point, Nasha, if the question is really just conveying or demonstrating the applicability across different types of mutations, it is important, definitely part of the goal of our drug. And that's why in Sue's lab, they took the time, the resources, the effort to actually go through and do multiple studies in multiple mouse models to demonstrate exactly that point.
Yeah, and I think this is such a, in the first couple of years of the foundation, we did a lot of fundraising and took a lot of time to create these mouse models. And I remember at the time we were thinking, why is it taking so long to build all these mouse models and then do the experiments and characterize the mouse models? But this is why it was so important, because we do have an array of mouse models and cell lines that go through different mutations. And this is exactly why we did it. So when we have a therapy, we can feel really certain that it could work for a variety of different mutations across the board. So that's why we spent all the time on that, the basic and preclinical side.
And just as important is having the clinical data from those mutations as well to
Yes.
layer over it. So at the same time as developing these assets of mouse models and cell lines, we were developing the natural history study and the patient registry. So we can really see the phenotypic, genotypic correlation, which, as we know, as we're getting to clinical trial readiness and our conversation with regulatory agencies, how important this data is, right?
Yeah, I mean, other foundations, other companies, they're honestly jealous of the work that we've been able to do, because without the clinical data, you can't, no matter how good your gene therapy is, you cannot take it to actually put into a human being and take to market. So all of the times we ask you to take a survey or, join the Citizen Natural History platform, the work is critical for us to get an actual drug for our children. So it's equally important.
Yes. And then the parent asks, what is the cost? What is the funds that we need to bring a gene therapy program through? And it's an estimated $22 million. And that's something to think about. So this is the cost of bringing a gene therapy through phase 1-2 clinical trials. Any therapeutic program afterwards is potentially the same cost. And this is what we're fundraising for. This is our lead program.
This costs money. So it is, the science is great. We could do all the science, all the clinical work, but without money, I don't know how to say that to everyone. We cannot do anything for our children. And something that I say to everyone is that it's not just my job or Nicole's job or, the foundation board's job. Each and every parent is all of our jobs to fundraise. And it doesn't matter if you fundraise $50 a year or $1 million a year, whatever you do, if we all do it together, we can push this through. And the beauty of our foundation doing the fundraising and pushing this drug through is that we are not beholden to any company and their investors and their priorities. Companies constantly drop programs, and as the wind shifts, we've seen this with companies, especially with the economic downturn. We have been able to actually push through because we've been able to raise the funds. So for all of you that are fundraising, and so many of you are, thank you so much, and we have to continue doing it. Please know that I hate fundraising just as much as the next person. So does Nicole, but it's something we all force ourselves to do because the price tag is high. But also, this price tag is not that high. Normally it costs several hundreds of millions of dollars. We've actually found ways to do things very efficiently as a foundation, but it is a team effort of fundraising.
Yeah. Yeah, Nasha and I just spoke at a conference really about this blueprint that we've developed to accelerate this timeline for us, the driven by parents, and that we've been able to cut the cost by more than half and cut the timeline by more than half. So as this parent-led and organization who's brought on the key people to work with us, like Gai and Elli, we can all feel good about that. We do not have to sit back and wait the time that it would, previously take to drive a rare disease drug to market. So again, yeah, thank you for asking that question about how much it will cost. And that is something that we are focused on every minute of the day. And as Guy could talk to, the timeline is very dependent on the funding. So, if we have a big bill to pay, then we can't push things forward if we don't have it in the door. We're confident we will be, but we need all hands on deck. I love this question, and it's a little repetitive because we spoke about it, but another question is looking at the time frame, best case scenario, when's the earliest that gene therapy will be in stage 5, which is patient clinical trials? And what she is referring to is our path to a cure on our website. Right, Nasha? I mean, we developed that years ago and it still holds up. Stage 5 is patient clinical trials, and we are in stage 4 right now of our path to a cure. How long will the clinical trial last before it is moved to gene therapy for the community? So, Guy, do you want to speak to that?
Yeah, so maybe there were two questions there that are related, but maybe a bit different. So the first one was the time that it will take for us to be in clinical trials. And as mentioned earlier, we think that if all goes well with the toxicology studies and with our regulatory interactions, then as early 2026, we can initiate clinical trials. Now, the second question is definitely harder to answer because it really speaks to when will a therapy be available to the community, which is another way of asking when will the drug be approved by regulatory agencies, which an approval means that it's no longer in clinical trials. Now it's a treatment that can be prescribed all over by clinicians. And the simple answer is that we cannot say yet, but I want to explain why. There are several factors that will determine this timeline, other than, of course, demonstrating that the treatment in fact works and it receives an approval, regulatory approval. The first is the duration. So to get to approval, we first of all need to get to start clinical trials, which we estimate will happen about 18 months from now. And then there is the duration of the trial itself, which is still to be determined. It will be most likely designed as a phase 1/2. Without getting into the details, it really sort of combines the safety and demonstration of efficacy in one trial. The duration of this trial, we have an estimate of when it's going to start. We don't yet have a good estimate of when it's going to end because the design of this trial is, as mentioned earlier, really what we're going to be diving deeper into with regulatory agencies and clinicians in the coming months. So to know when we're going to get to the finish line, we need to also first understand when are we going to expect this phase 1/2 to complete. Then there is the question of once this phase 1/2 is completed, traditionally, after this, regulatory agencies require what is called a confirmatory study, which is a phase 3, to demonstrate that what you saw initially in a phase 2 is actually, you can repeat that. This is usually how it's done. And of course, if we're required to run a phase 3, that means additional time and additional resources, quite a bit of them. So if the results of the phase 1/2 are robust and positive, then we could potentially seek what is called an accelerated approval from regulatory agencies to skip a phase 3. Now, there's no guarantee, of course, that regulatory agencies will agree to that, and it depends on the results that we get. And so whether or not one can file for an accelerated approval, which means that then it can get out to the community faster, greatly impacts when the drug is approved and can be, available to the broader community. So there are just multiple factors that we need to determine, and before we can give a better estimate. Maybe down the road we'll be able to give a little more granularity on maybe if we get an accelerated approval, then what's our estimate, and if not, then what's our estimate. For that, we'll need to finalize our phase 1/2 design, and then we may be able to communicate some options. But it's hard to do before we're having these dialogues with clinicians and agencies. So stay tuned.
Great. That's a really helpful answer to manage expectations. Oh, you know what? I'm going to skip ahead a little bit what we're all looking at here, because I know, Elli, we're going to lose you on this Zoom in about 10 minutes. So I want to ask a question here that this one's outside of the gene therapy topic, but a mom pointed out that she finds parents are sometimes confused about their mutation that their child has, especially when the word dupe is in the name. Sometimes I think it's a duplication. One parent, she was pointing out, this is actually a frame shift. So can you give, well, first we will say that at the conference, Elli will be doing a session called Ask Elli. So if you bring your genetics report or you bring your child's mutation, Elli will break it down for you. And she will talk about, what does this mean? How do you know? And based on all the clinical data that we have, what do we know about it? So when you come to the conference, you will have your own session with Elli. But in the meantime, to answer this question, she wants to know, are there any tips and tricks you could answer right now to help parents sort of understand their mutation a little better? And we've done this on our YouTube channel too.
It's a great question. And I was going to say, perhaps we can provide the link when we post this video to the YouTube, because it is a lot to cover quickly. I will say one thing that could be helpful when we're thinking about mutation class is sometimes when you look at your genetic test report, the same FOXG1 variant will be described in 2 different ways. So there will be one way that is describing the variant in the DNA sequence, and you can tell that that is the DNA sequence because it starts with a C dot. So for example, in this question, C.460 dupe, that's one of the more common FOXG1 variants. But it will also be described in a second way, which is looking at the amino acid sequence, or what the impact of the variant is on the protein. And you can tell that that is the protein because it's preceded by a P dot. So I believe the C.460 dupe would correspond with a P dot glue 154 Gai, I want to say. Don't quote me on that, but I think it's pretty close. So basically it's the same variant, but looking at either the DNA sequence or the protein sequence. And oftentimes the clues or the tips or tricks in classifying the mutations are actually in the protein sequence and not in the DNA sequence. So for example, for all of the frame shift variants, in the protein sequence, you'll actually see the letters FS, and it's like an acronym for frame shift. For all of the nonsense variants in the protein sequence, you'll see a little asterisk, and that is a symbol for a nonsense variant. And then in the missense variants, typically what you would see is either 3 letters or 1 letter, a number, and then 3 letters or 1 letter. So it's 1 number sandwiched by letters on either end. So those are kind of the tips or tricks that I look for, but as Nicole mentioned, happy to go in on detail for specific variants of interest at the conference, and I happily answer emails about this as well. So please don't hesitate to reach out if you have specific questions.
And I will post the link to our YouTube video where you gave a webinar on deconstructing your genetics report.
Why do they say dupe when it's not a dupe followed by the C dot?
Because it's a duplication of a single DNA nucleotide. So it's the number 460 dupe means that nucleotide or DNA letter 460 is duplicated. So it's that extra letter that creates the reading frame shift, essentially. I know it's confusing.
That's why we
You
have
made-
Elli. I can't wait for our Ask Elli session conference. And while you're here, there's another question I think that would be perfect for you to answer.
Sure.
And this is actually from a mom in Turkey who wanted to know, do other children with FOXG1 have scoliosis and contractures in their legs? And we know what we know from our natural history study and registry.
Yeah, I think this is such a great plug for participating in some of the clinical research programs. Characterizing these phenotypes or symptoms, as well as how commonly they observe in the population, are one of the main aims of our study. So we certainly do see scoliosis and contractures in the legs and the arms and the hands in FOXG1 syndrome. And this is specifically true as people get older. And when we saw this question, we went back and looked at it in the NHS data that we have available today. We have data collected for about 101 individuals with FOXG1 syndrome. And the frequency of scoliosis was about a quarter. So almost 25% of those 101 individuals had a diagnosis of scoliosis in particular. And importantly, the average age of that cohort was 9 years. So one thing that we could think about doing in our analysis that we share back with the community in November is looking to see whether any of these symptoms change in frequency with different age groups as well. So it was a great question and a great prompt for us to continue to think about the ways we can investigate our data.
Yeah, and it actually, I want to piggyback another parent's question who said, and this is going back to our clinical trials, but how can parents help with outcome measures? So that's a great question. And thank you so much for asking that, because it's so important that every parent, that we know what the outcome measures are. So I'm going to let you, Elli, touch on that, the importance of these studies and how parents can help with outcome measures.
Yeah, I would say that, and just to clarify for everyone, when we talk about outcome measures, what we're referring to are what are the signs or symptoms that we could measure in a clinical trial that we might expect to change when someone receives an investigational therapy like a gene therapy. So a lot of the clinical work that we are funding right now is to better understand not only what are the common signs and symptoms of FOXG1 syndrome, but what is most important to families. So I kind of like to think of it as 2 different arms. We have things like our patient registry and our natural history study, which are really intended to provide a very detailed and comprehensive overview of FOXG1 syndrome across the lifespan. So what does FOXG1 syndrome look like in infancy? How does that change in childhood? How does that change for adults? Are there variations between people with one type of mutation versus individuals with another? So kind of providing this really rich perspective on an objective perspective on what FOXG1 syndrome is, so that we can be thoughtful about how we choose these outcome measures. And then the second arm that we have is, I work very closely on our natural history studies, so I'm certainly biased, but it's such an incredible resource. But you lose a little bit of color in that we can tell you what is most common. We can tell you what medications people are taking, but the data doesn't necessarily give the patient perspective or the caregiver perspective. And that is really important to us, that we make sure we're designing clinical trials that actually measure what is most important to you. So Q, our disease concept model, and the interviews that parents have been participating in. We have another exercise that we'll be recruiting for soon, so stay tuned. But that is really intended for us to complement this kind of objective data with the parent perspective and saying, this is what is most meaningful to my child. This is what would make the biggest impact in my life, and making sure that that perspective is being incorporated in clinical trials. So I think what everyone can do is, as your time allows, we certainly respect that everyone is very busy, but as you are able, the more you're able to participate in these studies, the more we're able to really improve our understanding of FOXG1 syndrome and make sure that those insights are translated into our study design.
Yeah. And I think parents do get a little confused and overwhelmed sometimes, like, wait, which study am I supposed to do? What are the difference of all these studies? And that was a really good explanation of the natural history study, what that's the purpose for it, and the disease concept model, the difference of that. But I do want to say at the very beginning, the first step for parents to do is just registering your child as having FOXG1 syndrome. And that's right on our website under the parents and caregivers. It says patient registry, and that's really just like the first step. But this is the official number of FOXG1 patients in the world. And that is so critical that every FOXG1 patient is accounted for. Nasha, you could speak to a little bit why, we're always saying, is your child registered? Is your genetics report updated? And what it means to pharma, what it means for us, for the epidemiology of FOXG1.
Exactly. There is no other database. If you think the hospital systems are connected and there's some way for everyone to know how many FOXG1 kids there are in the world, that does not exist. So truly what Nicole said, our registry is the official platform to count FOXG1 children, patients. And when pharma companies think about which rare disease they're going to develop a drug for, they actually contact foundations like ours and ask us what our count is. And this is why it is so critical. And it's also critical to help all of us, even if you don't believe in a cure. Okay, let's say you're like, I don't believe a cure is possible, but we all need to manage our children day to day. They're all having symptoms that we need to manage. And all of us coming together and sharing the experiences that our children are going through help all of us manage our own children as we're waiting for these therapies to come down the pipeline. So it is, if you do nothing else, please just register for the patient registry. It does not take a long time. And then if you can sign up for the natural history study, if you're located in the United States, that's amazing. If you can join any of the other projects that Elli or Nicole asks, please do that. And also know that we get asked by many organizations to send surveys out to you. And we're very careful to only send things that are going to actually help us in our path to a cure and help us as a community. So please know that we are very mindful of your time and our own time. We're all parents as well. We're very mindful of our time. And so we do just ask what is absolutely necessary to help all of our children.
Yeah. Starting with the registry, just registering your child for the greater good of FOXG1 syndrome. It's for every other parent and every other patient out there. It could be somebody wants to know, well, how many patients are there in Turkey? We should be able to know. But if there's parents in Turkey who didn't register their child, and it's also going to be very important when we come to the time of clinical trials to know
Yes.
who's out there. So Elli, did you have to jump off?
I do, but thank you so much for all of your wonderful questions, and I'm so excited to share more with you at the November conference.
Thank you, Elli. Thanks. Okay, so we're going to go backwards in this list of questions we have because I jumped ahead. And this was a really great question from a mom here in New York as well. And she asked, is there an age where treatment will no longer be beneficial? And I know that a lot of parents with older children are wondering this. So Guy, do you want to speak to that?
Yeah, this is definitely a question that is front and center in neurodevelopmental disorders in general, including FOXG1. And I think the best answer that we can give is that the only way to know for sure is to test this in clinical trials. However, there is out there, when I say out there, I'm talking about the discussion in the scientific community, this notion of what is called the therapeutic window, or sort of an age range where treatments may work and outside of that may not work. And so maybe I'll just say a couple of words about that. I think that this notion comes from the thinking that the brain develops up to a certain age, and then after that, maybe it's harder to make changes. However, what we are starting to see from programs for other conditions in the clinic is that this may not really be the case. Take, for example, recently, information that came from an Angelman syndrome trial where teenagers that have been on an antisense nucleotide treatment are showing really impressive treatment effect. Rett syndrome, Tasha's trial, with an older patient seeing signs of potential treatment effect. All these are anecdotal at this point in time. We do not have a treatment that made it to the finishing line and got approved such that we have the entire data set. What we have is bits of information from ongoing trials, but they're certainly encouraging and suggest that maybe this idea of a window of intervention is, we can't say at this point in time if it doesn't exist or does exist, but maybe it's not as clear cut as initially thought. And a lot of the thinking comes from preclinical studies, and there it's just important to recognize the limitations of animal models. We need them. They help them a lot. They help us a lot in the development process of therapies, but they are limited, and not everything that is predicted in a mouse model will translate into humans. And so specifically for FOXG1, we will not know until we're in the clinic. However, at this point in time, I would say that we do not have any information that would suggest that there is a well-defined window or age range beyond which we will not see a treatment effect, right? That does not promise that we'll see a treatment effect in all ages, and maybe it'll be differential between ages. We won't know. I think that it is also worthwhile mentioning that, again, I'll bring up the amazing work that is done in the Lee lab. They have looked at older mice, disease model mice, where they treated them with our candidate gene therapy and demonstrated treatment effect even in older mice, such that, again, with the limitations and caveats of the preclinical models, I think that I see no reason to conclude prematurely that older patients will not be able to benefit from this treatment. So that's what we have clinical trials for.
Which is hopeful. I know when the Lees got the data from the older mice, there was a lot of excitement about it, and of course, you putting in the disclaimer that, well, these are mice, and but it is very exciting to see that data versus seeing the opposite.
Yeah, and I think it's really exciting to see all the other clinical trials that are ongoing for Rett syndrome, for Angelman, for Gai-1, and patients that are getting treated are older and they're seeing actually efficacious results. So this is amazing.
Yeah, absolutely. Okay, so on back to our roadmap, our gene therapy roadmap, we talked about being in the toxicology phase now, and one mom wanted to know a little bit about the toxicology studies. When did they start? How long did they last? And I think what's a really interesting question is what happens if toxic responses are observed? Does it
Yeah.
delay the treatment development? Are there ways to modify the therapy to be less toxic? How reliable are studies in non-humans when it comes to toxic reactions? And what she said, this is interesting because if the gene therapy is a one-time treatment, how can you really safely trial it?
Yeah.
Well, that's important to answer.
Yeah, no, all fantastic questions.
Mhm.
I'll start by saying that the toxicology and safety studies, are mandated by the regulatory agencies, and the studies that we do they have to be in line with the agency's requirements. In other words, we're following, this is where we're entering a regulated territory, right? And we're following the guidelines of the regulatory agencies. In terms of toxicity, and this is a general statement that's not specific to gene therapy, even drugs that are taken over the counter every day, like aspirin or Tylenol, if you take too much of them, you'll have toxicity. So any drug given at the right dose can be beneficial, and at the wrong dose have toxicity. The purpose of toxicology studies is to understand the relationship between dose and toxicity. Now, from the work that we have done and Sue and her team has done at University of Buffalo, we have the understanding of the relationship between dose and efficacy, right? So a treatment effect. Now we're going to study in toxicology studies the relationship between dose and toxicity. And what you want to see and what you see in many drugs is what we call the therapeutic window, or what doses do we need for efficacy, what high doses we're starting to see maybe toxicity or adverse events, and then that gap between them, right, that margin between them is really the therapeutic dose. This is the dose where you project that you can dose safely and get efficacy. So the purpose of the toxicology studies is really to help us define that range or that we call the therapeutic window. This helps then, in discussion with the regulatory agencies, define the doses that will be tested in a clinical trial. Now, sometimes with some drugs, and it happens more times than not, one can run in the toxicology studies into a problem where you see that the dose where you see toxic effects is too close or overlapping with the doses that are needed for efficacy. So if that happens, it really means that you do not have a safe drug, and you need to go back to the drawing board and understand why there is toxicity and fix it. So as long as one sees in the toxicology studies that indeed there is a good enough window, there is a margin where one can dose safely for efficacy but stay away from toxic levels, that really then, in alignment with the regulatory agencies, gives you the opportunity to test this in humans in the clinic, and that determines the doses that will be tested in the clinic. Okay, I hope this makes sense. So when are we going to go back to the drawing board or look about, if there's toxicity, do we fix it? Well, it depends on whether or not we have that window. If we have that window, then, we should have a path forward. If not, then we need to understand why we do not have, what is the source of toxicity, fix it, then go back. So this is why when we were asked earlier about the time to start phase 1/2, well, it'll be early 2026 if the toxicology studies show us that we have that window to go into the clinic with what is projected to be a safe dose. If not, then it'll take a little longer.
That was a great answer, Guy. It also speaks to what does this cost? Well, there's a lot of ifs. A good thing that toxicology is at the beginning of the roadmap, so that if you go back, you're going back to your compound, right? But I hope that answers the question. Do you have anything to say on that, Nasha? Any,
No,
Mhm.
I mean, I think exactly what you said. This is why it's really hard to exactly give a number for what drug development costs. The numbers that we've given, 22 million, are a best case scenario, and we truly hope everything goes well. But, this is the reality of drug development and of science. You don't know what you don't know.
Yeah, and I guess parents want to feel comfortable that if they're putting their child in a clinical trial, that it will be safe.
And
So this toxicology phase, is it foolproof? Do we know for sure this is not a toxic compound? When we're in that toxicology phase, they want to know that, there's a reason for it. And we know that in human clinical trials, there's not going to be an adverse effect, right? Or a toxic outcome.
and that is the entire point of toxicology.
And this is why this is a regulated space, right? This is why you actually need alignment with the regulatory agency before you are enabled to take it into clinical trials. It is important, I think, to just state that nothing is foolproof, right? So basically, you do the best you can based on very strict and clear guidelines from the agencies. And with that, you move into the clinic. But even then, when you start your clinical trials, you start at a lower dose and you work your way up, such that even when you're in the clinic, it's not that if you pass the toxicology studies, then happy days and you roll into the clinic and that. No, even then, the clinical trial is designed in such a way that you make your way up the doses in a monitored and safe way. And if you detect a safety signal that was not observed in animals, then you see it at low doses rather than going high, right? Right off the bat. So even once you enter into the clinic, safety is still the priority. It's the priority preclinically, and then it continues to be the priority clinically.
Yeah, absolutely. There's something to be said about being a parent-led organization with the Lees being parents themselves, where, safety is of, highest concern. But like you said, it's a regulated industry. So the last question is, when the time, how and where would I be on the list for clinical trials when they become available? So what we'll say, when the time comes, your best resource is staying in touch with us. Oh, first of all, make sure that you are getting our emails. There's a form on the parents and caregivers on the newly diagnosed page. Fill out that form to get our emails, our newsletter. If you're on social media, make sure you're following us on social media. You have, how else? Just be in touch with us. Look at our website, and every parent will be informed when there's clinical trials. For clinical trials in general, there's clinicaltrials.gov, but for the FOXG1 community, being in touch with us is going to be your resource, your best resource to know what is, when the time comes.
Agree.
Great. Yeah. So again, we hope that you will join us in November at the FOXG1 Parents Conference in Pompano Beach, Florida. It is such an amazing experience. We did it there 2 years ago, and just all being together and having deep conversations is far beyond anything that we could do here. Our whole team will be there. There will be leading neurologists in FOXG1, as well as many exciting guests. So we hope that this Q&A session was helpful for you. Again, reach out to us anytime. We are here for you. We are your foundation. We are FOXG1 syndrome parents, and we're just so happy to have the ability to share this with you, and we thank you for doing your part on this journey with us.
Yep. Thank you.
Thank you.
Thank
Thank
you.
you, Guy, for this incredible explanation.
Thank you.